There's a dish of seawater sitting on a bench in a lab on the Italian coast, and the animal inside it is doing something wrong. It's a jellyfish, though calling it that oversells the size. The bell is about 4 millimeters across, less than a quarter of an inch, which means the whole animal would sit comfortably under your little fingernail with room left over. Through the transparent bell, you can see a bright red stomach, and around the rim, a fringe of tentacles that fold and drift. This is Turritopsis. In the late 1980s, a German biology student named Christian Sommer collected some of them in the water off Rapallo, on the Italian Riviera, and brought them back to keep in dishes, the way you do when you're young and someone has finally let you loose on a microscope. And the medusae in his dishes started dying. That's normal. That's what medusae do. A jellyfish bell is the last chapter of a hydrozoan life. The animal spends most of its existence as a polyp, a tiny stalk cemented to a rock or a shell or a piece of drifting plastic, and at some point that polyp buds off little swimming bells, and those bells drift and feed and release eggs and sperm, and then they fall apart. The bell goes cloudy, the tentacles shorten, the whole thing contracts down into a lump of sagging jelly and sinks, and within a day or two there's nothing left in the water but a smear. Sommer's medusae did the first half of that perfectly. They contracted, they sank, the tentacles pulled in, and the bell went from a clean glass dome to a shapeless blob on the bottom of the dish. Then, the blob stuck to the glass. And over the next few days, it grew a stolon, a creeping root-like thread. And out of that thread came a polyp, which is the wrong direction. That's the equivalent of a frog turning back into a tadpole or a butterfly folding itself into a caterpillar. In animals with a life cycle like this one, the arrow only points one way, and everybody who works on hydrozoans knew it pointed one way, and here it was, in a dish, pointing the other way. Giorgio Bavistrello, Sommer, and Michele Serra put a first report of it into print in 1992. 4 years later, Stefano Pirino, Ferdinando Boero, and their colleagues published the full account in the Biological Bulletin, under a title that doesn't waste anybody's time: Reversing the Life Cycle. Medusae transforming into polyps. You've almost certainly heard about this animal. It got a nickname, the immortal jellyfish, and the nickname did what nicknames do, which is eat the actual biology and leave a slogan behind. I'm not here to sell you the slogan. What interests me about that dish in Rapallo isn't the word immortal at all. It's a much more awkward problem sitting underneath it, and once you see the problem, you can't unsee it. To say that an animal cheated death, you have to know what death is. And here's where it gets uncomfortable, because we all act like that's settled. Death is one of the very few things every human being is supposed to understand without instruction. We build law around it. Medicine, insurance, religion, inheritance, grief. All of it rests on the assumption that there's a line, that the line is real, and that we know roughly where it sits. Biology does not have that line. Biology has a pile of candidate lines, 6 or 7 of them, depending on how you count, and they were drawn by different people solving different problems at different scales, and they contradict each other. Most of the time you never notice, because most of the time the candidates agree. A gull hits a window and every definition you could reach for says the same thing at the same moment. Metabolism stopped. The body's failing. The cells are going. The individual is gone. When all your instruments point the same way, you don't find out that they measure different things. The ocean is where they stop pointing the same way. Not because the ocean is magic, because it's old and enormous, and it contains lineages that split off from ours before most of the assumptions in our definition of death had been invented. Sponges and jellyfish and sea cucumbers and clams and worms went their own way somewhere between 500 and 600 million years ago, and they've spent every year since then solving the problem of staying alive under conditions we'd consider a formality. No oxygen, no water, half the body missing. 4 centuries of continuous existence in the same square foot of mud. So the honest version of the question in the title isn't how these animals cheat death. Cheating implies a rule, and a rule implies somebody wrote it down correctly. The version I actually want to chase is this. Take each of our definitions of death, one at a time, the ones we actually use. Find the marine animal that walks straight through it, and see what's left standing at the end. I'll tell you now that something is left standing. There's a definition that survives contact with all of them, but it's much, much smaller than the one you're carrying around, and by the time we get there, I think you'll find it's changed shape on you. Let's start with the one the jellyfish breaks, because it's the oldest and the most intuitive, and almost nobody says it out loud. Death is the end of a one-way trip. That's the shape of it. Not the mechanics, the shape. You're born, you develop, you mature, you decline, you stop. The order is fixed. You can move faster or slower along the track. And medicine has gotten pretty good at slowing you down in places, but you cannot turn around. Every culture that's ever thought about this has thought about it as a journey with a direction. Rivers running to the sea, sand through the glass, candles. The metaphors are all irreversible processes, and they're all irreversible because we've never had a reason to imagine otherwise. Turritopsis dohrnii, which is the species that turned out to be doing the party trick, ignores the direction. Now, how? This is where I have to slow down because the mechanism is more interesting than the headline. And it's also where the honest limits start showing up. When a Turritopsis medusa gets into trouble, and trouble here means physical damage, starvation, a sudden temperature swing, being poked with a needle by a graduate student, it doesn't just wilt, it executes something. The bell begins to contract, the tentacles retract and get resorbed into the body. The mesoglea, the jelly, thickens and clouds. Within somewhere between 1 and 3 days, depending on temperature, the entire animal has collapsed into a small round cyst, and that cyst settles onto a hard surface and glues itself down. From the cyst, stolons creep outward. From the stolons, polyps rise. And those polyps, given food and time, will bud off new medusae, which are genetically identical to the one that collapsed. The part that made cell biologists sit up straight is what the cells are doing during that collapse. They're not dying and getting replaced from a reserve of stem cells. That would be strange enough. They're changing job. A cell in the striated muscle of the swimming bell becomes a cell in the epithelium of a polyp. A cell that was doing one specialized thing, with one specialized set of genes switched on, shuts that program down and boots a different one. The technical word is transdifferentiation, and the reason it's a big deal is that in most animals, most of the time, specialization is a door that locks behind you. Once a cell has committed to being a muscle cell, it stays a muscle cell, and it will divide into more muscle cells, and eventually it will die as a muscle cell. Turritopsis now walks back through the locked door and comes out as something else. Pireno's group reported that under the right conditions, essentially every stressed medusa, they had made the transition. This is a capacity the animal carries around with it, sitting there, waiting for a bad enough day. And then Shin Kubota happened. Kubota is a marine biologist at Kyoto University's Sato Marine Biological Laboratory in Shirahama, on the Pacific coast of Japan. And he has spent decades keeping these animals in tanks and feeding them by hand, which mostly means brine shrimp delivered with a pipette, one medusa at a time, for years. In 2011, he published a short note in the journal Biogeography, reporting that he'd watched a single Turritopsis go through the reversal 10 separate times over about 2 years. 10. Bell to blob to polyp to bell, 10 times. And the 10th polyp looked like the first polyp. I want to be honest about what that paper is, though. It's a 3-page observational note from one lab on animals in tanks. And it's the strongest published record we have of repetition, which tells you something about how thin the ground is under the word immortal. Nobody has followed one genetic lineage of these animals across a human career and demonstrated zero degradation. Nobody has done the experiment that would settle it, partly because it's a brutally hard animal to keep alive, and partly because the experiment would take longer than a career. In 2022, a Spanish team led by Maria Pascual Torner, working with the group of Carlos Lopez Otin at the University of Oviedo, went at it from the genome side. They sequenced Turritopsis dohrnii and, crucially, they also sequenced Turritopsis rubra, a close relative that does not do the reversal. Two species, same genus, one trick between them. That's about as clean a comparison as this kind of biology ever gives you. What they found was a set of differences clustered in exactly the places you'd guess. Genes for repairing damaged DNA, genes for maintaining telomeres, the protective caps on the ends of chromosomes that shorten in most animals as cells divide, genes handling the cell's oxidative chemistry, and genes governing the stem cell population. The immortal one had roughly twice as many copies of some of the repair and protection genes as its mortal cousin. During the reversal itself, the transcriptome showed a particular set of genetic switches, the polycomb repressive complex targets, going quiet, while pluripotency genes, the ones associated with cells that haven't decided what to be yet, came on. That is a satisfying result, and it deserves its due. But notice what it is. It's a correlation between a set of gene variants and a capacity. It shows you the toolkit. It does not show you the animal using the tools step by step, in a way that would let you say this gene causes that reversal. The authors were careful about that. The coverage mostly wasn't. So where does that leave the first definition? The medusa is destroyed. Let's be clear about that, because the slogan skips it. The bell, the tentacles, the swimming animal that was drifting around eating copepods. That thing ends. Its body is dismantled. If you were watching one particular medusa because you'd grown fond of it, you would watch it stop existing. What continues is the cell lineage and the genome, reassembled into an earlier form, and then that earlier form buds off not one medusa, but several. So the thing you were following becomes 4 things, or 12. Ask which one is the original, and the question falls apart in your hands. There isn't an original, there's a branching. They also get eaten, constantly. Turritopsis medusae are food for anything that filters the water column, and a polyp cemented to a dock piling is food for sea slugs, and none of this genomic sophistication does a thing about being digested. In the wild, these animals die at ordinary rates from ordinary causes. What they don't do is run out of turns. So the first definition doesn't survive, and it fails in a particular way that's worth naming, because the same failure is going to happen again and again over the next couple of hours. It fails because the definition was smuggling in an assumption that wasn't about death at all. It assumed that a life is a trajectory with a fixed direction, and that assumption turns out to be a description of us, and of frogs, and of oak trees, and not a description of animals in general. Draw a line and something swims through it. So we try a firmer definition, something with less philosophy in it. Death is what happens when the body is destroyed. That one feels solid. It's the definition every accident report uses and every coroner works from, and it has the great advantage of being physical. Forget trajectories and life cycles. There's a body, the body is a machine, and when the machine is broken past the point where it can run, the animal is over. You can point at the moment. You can hold the evidence. There's a story that circulates in fisheries history, and it's told often enough that it's worth telling carefully. Oyster fishermen along the American Atlantic coast, working beds that sea stars were eating their way through, would haul the sea stars up, chop them in half to be sure of the job, and throw the pieces overboard. And the population, according to the story, went up. I can't tell you that specific account is documented the way I'd want it documented. Old fisheries lore is full of stories that hardened into fact through repetition. What I can tell you is that the biology permits it in some species under some conditions, and that the reason it permits it is one of the strangest facts in animal anatomy. Start with the ordinary case. Most sea stars can lose an arm and regrow it. That's already unusual by our standards, but it's the direction you'd expect. The animal keeps its center and rebuilds a limb. The severed arm, meanwhile, sits on the seafloor and rots. Now flip it. There's a genus called Lynchia, tropical sea stars, often blue or a mottled red, common on reefs across the Indo-Pacific. Lynchia does something that shouldn't work. An individual arm detaches, deliberately, at a soft point in the body wall, and it crawls away. Not as debris, as an animal. It has no mouth. It has no central disc, which is where the stomach lives. It has no way to eat anything at all. And it walks off across the reef on its tube feet, living entirely on the nutrients stored in its own tissue. And over roughly 10 months, it grows a new central disc, and out of that disc, 4 new arms. They come in small, maybe half an inch long at first, so what you get is a full-size arm with a little cluster of stubs at one end. Divers call them comets, because that's exactly what they look like, a long body with a short, bright tail. Meanwhile, back where the arm came from, the parent sea star has regrown the missing limb and is going about its business. Count the animals. There was 1. There are now 2. Neither of them died, and no reproduction occurred in any sense a biologist would recognize as sexual. And one of those 2 animals contains a chunk of tissue that was, 10 months ago, a limb. In Lynchia multifora, this happens so routinely that it's classified as a normal mode of reproduction for the species. The animal reproduces by coming apart. I keep trying to find the moment in that sequence where the definition applies, and there isn't one. The body was destroyed, in the sense that the original arrangement of parts stopped existing. Nothing about that destruction resembles death. It resembles division. Then there's the group that takes this further than anyone, and takes it in a direction that made me put the paper down for a second. Sea cucumbers. Holothurians are, to be blunt, not the charismatic end of the ocean. They're soft sausages on the seafloor, they eat sediment, they process an astonishing volume of sand, and most of them look like something that fell off a truck, and they can throw out their own internal organs. The word is evisceration. Under stress, and the documented triggers include crowding, low oxygen, sharp temperature swings, and rough handling, a sea cucumber will expel its digestive tract, its respiratory trees, and in some species a substantial fraction of everything else in its body cavity. Some species do this out the back, some do it out the front, tearing open the front end of the body and ejecting the gut through the mouth. The animal ends up as essentially a tube of body wall, with a nervous system, lying on the sediment, empty. That should be the end of the sentence. It isn't. The mechanism that lets them do it is called mutable collagenous tissue, and it deserves more attention than it gets. Connective tissue in a sea cucumber sits under nervous control. Specialized cells release molecules into the collagen matrix that change how strongly the fibers bind to each other, and the tissue goes from tough to nearly liquid in a matter of seconds, at specific planes, on command. That's how the body wall opens, without tearing randomly. There's an autotomy plane, and the animal softens it, and the organs come out. Then it rebuilds them. In Opisthocoppus japonicus, the Japanese sea cucumber, which is farmed commercially and therefore extremely well studied, the intestine regenerates as a fully connected tube with an open lumen in about 14 days. 14 days from empty body cavity to working gut. The immune cells circulating in the body fluid are back to normal counts by around 35 days. Overall physiological recovery, by the measures used in the aquaculture literature, lands somewhere near 45 days. The gut bacteria take longer, several months, to reassemble into a stable community, which I find quietly hilarious. The animal can rebuild its own digestive tract faster than it can rehire the bacteria to run it. So run the definition again. Was the body destroyed? Most of the working parts of it were on the seafloor. Was the animal dead? It was alive the entire time, and 45 days later you could not tell by looking. At this point, somebody usually says these are simple animals, and simple animals do simple things, and we shouldn't be surprised. I want to push back on that, because the assumption underneath it is wrong twice. First, echinoderms are not simple. They're deuterostomes, same broad branch of the animal family tree as us, and their nervous systems and immune systems and developmental genetics are elaborate. Second, and this is the part that undermines the whole objection, the same trick shows up right next door to the vertebrates. Botrytis schlosseri is a colonial tunicate, sometimes called the star ascidian. You've probably seen one without registering it. A flat crusty patch on a dock piling or the underside of a boat, patterned with little star-shaped clusters. Tunicates are chordates. They're our closest invertebrate relatives. Their larvae have a notochord and a dorsal nerve cord, the same architecture that our own spine descends from. And then the larva settles, resorbs its tail, and becomes a filter-feeding blob, which is one of evolution's better jokes. Here's what Botrytis can do. Take a colony and remove every single one of the zooids, the actual bodies, the parts that eat and filter and reproduce. Leave nothing behind but fragments of the vascular network, the tubes of shared circulatory tissue that connected them, with the blood cells inside. There is now no body in the colony, none. Within a week or two, a complete new adult body regenerates out of the blood vessels. Not a bud from a surviving zooid, because there aren't any. Cells circulating in the vasculature aggregate, reorganize, and construct a whole functional animal with a gut and a heart and a filter basket starting from what amounts to plumbing. Work on the onset of that process points to intense remodeling of the vessels in the first day, with genes for building new vasculature and rebuilding the extracellular scaffold going up hard. It's whole body regeneration in an animal on our own branch of the tree. And then there's the worm that finally made this into something you can test. Hofstenia miamia is a marine acoel, a flatworm-shaped animal a few millimeters long that lives in shallow sediment around Bermuda. It's known as the three-banded panther worm. Cut one into pieces, and each piece builds a complete new worm, head and all, in about 8 days. Planarians have been famous for this since Thomas Hunt Morgan was slicing them up in the 1890s, before he moved on to fruit flies and won a Nobel Prize for something else entirely. But planarians and acoles sit far apart on the animal tree, which makes the shared ability an open and rather good question. What Mansi Srivastava's lab at Harvard did, with Andrew Gehrke as first author, was work out how the machinery is wired, and they published it in Science in March of 2019. Their approach was to look at the physical state of the genome rather than at the genes alone. DNA in a cell sits wound and tightly packed, and packed regions can't be read, so they mapped which regions of the genome unpack after a wound and when. Thousands of regions open up. Then they looked for the common signature in those newly opened regions and found the binding site for one gene, early growth response, EGR. EGR turns out to be sitting at the top. It acts as what's called a pioneer factor, which means it can bind to DNA that's still packed away and pry it open, and once it's open, the rest of the wound response program can be read. Knock EGR down with RNA interference, and the downstream genes never fire, and the worm doesn't regenerate. It just sits there with a wound. I want to flag the difference between that result and the jellyfish genome I described earlier because it matters, and this is the kind of distinction that gets flattened in retellings. The Turritopsis work compared two genomes and found gene variants associated with a capacity, association. The Hofstenia work removed one gene's function and watched the capacity disappear. That's a causal test. Both are good science, and they're not the same weight of evidence. And anyone who tells you a genome comparison explains a phenomenon is skipping a step. So the second definition is in trouble, and I think it's worth naming exactly why. Because the failure isn't about regeneration at all. Bodies as the unit of life is an idea we got from having a particular kind of body. Ours is centralized. One heart, one brain, one blood supply, one set of plumbing with essentially no redundancy in the places that matter. Damage in our bodies propagates because everything depends on everything. And the failure of one component takes down the system. That's not a rule of biology. That's an engineering choice, made a very long time ago in our lineage, and it came with trade-offs we're still paying. An animal built out of modules, with distributed control and stem cells scattered through its tissues, and connective tissue it can dissolve at will, has a completely different relationship to physical damage. Cut it, and you have not damaged a system. You have subdivided one, which means the body was never the thing. The body is a vehicle the thing is currently driving, and some animals keep several sets of keys. Fine. Third attempt, and this time we go for something you can actually measure with an instrument. Death is the cessation of metabolism. This is the grown-up definition. It's the one physiology textbooks lean on, and it has real content. Life is a set of chemical reactions running against the current, holding molecules in arrangements they wouldn't hold on their own, paid for by a constant supply of energy. Stop paying, and the arrangements relax. Everything falls downhill. That's death, described in thermodynamic terms, and it's elegant enough that when I first learned it, I thought the argument was over. For animals, the energy comes from oxidizing food, and the reason oxygen is involved is almost embarrassingly practical. Breaking a molecule of glucose apart releases electrons, and those electrons have to go somewhere, and oxygen is an outstanding place to put them. It's greedy, it's abundant, and the drop in energy as electrons fall onto it is enormous. Run that process fully, with oxygen at the end of the chain, and a molecule of glucose yields somewhere around 30 units of the cell's energy currency, a figure textbooks argue about, and which varies with conditions, so treat it as a rough number. Run it without oxygen, with only the first few steps of the pathway, and you get 2. 2 versus 30. That gap is why animals breathe. And in us, the gap has teeth. Our brains consume about 1/5 of our energy budget while making up around 2% of our body mass, and they hold essentially no reserve. Interrupt the supply, and the failure begins in seconds and becomes irreversible in minutes. That number, those few minutes, is quietly built into every definition of death that any human institution uses. We drew our line where our own chemistry breaks. Go down to any rocky shore at low tide and you can watch a whole community ignoring it. A mussel bed exposed to air is a mass of animals with the door shut. Blue mussels clamp their shells and wait, and while they're waiting, there's no water flowing across the gills, which means no oxygen coming in, which means the whole aerobic apparatus is offline. Twice a day, every day, for their entire lives. And in cold water, if conditions demand it, they can extend that from hours into days, and in some experiments, weeks. There's more going on under that shell than held breath. The chemistry down there is different from the one we fall back on. When our muscles run short of oxygen, we do the simplest possible thing, glycolysis, ending in lactate. It's fast, it's poor, and it comes with a penalty, because you're piling up an acidic end product inside yourself with nowhere for it to go. That's why the human anaerobic option is measured in seconds and minutes. Bivalves and marine worms took a different route. They run coupled pathways, burning stored glycogen alongside amino acids like aspartate, and their end products are things like succinate, succinate, propionate, acetate, and alanine, plus a family of compounds called opines that do the same job as lactate without the same acid load. It's slower. It's still far worse than breathing, but it yields noticeably more energy per unit of fuel than our lactate route. It acidifies the tissue far less, and it can be sustained. The lugworm buried in the mudflat, Arenicola marina, does this for hours at a stretch as the tide goes out and its burrow water goes stale. The peanut worm Sipunculus nudus became a laboratory model for long-term anaerobiosis, precisely because it will tolerate conditions that would end most animals before lunch. Then there's the part of the ocean where this stops being a tidal inconvenience. Across large stretches of the eastern Pacific, there are layers of open water, hundreds of feet thick, where oxygen is nearly absent, and the Humboldt squid drops into them every single day. It's a big, fast, muscular animal that works the productive surface layers at night and spends the daylight hours down in that layer. Rui Rosa and Brad Seibel, publishing in the Proceedings of the National Academy of Sciences in 2008, measured what the descent does to it, and the answer is that it turns itself down. Under those conditions, their measurements showed its metabolic rate dropping by around 80%. A big, powerful animal voluntarily cutting its own engine to a fifth of normal for hours at a time, then coming back up and switching it on again. I find that one hard to file. It's an animal treating its own metabolism as a setting. The clearest worked example of the whole strategy, though, isn't marine at all, and I'd rather tell you that up front than quietly let you assume otherwise. It's a fish in the ponds of northern Europe. The Crucian carp spends winter under ice, in small ponds where the water goes completely anoxic for months. Not low, 0. And the carp is fine. It stays awake, it moves around. Its brain keeps working, and when the ice melts in spring, it swims off, having spent the better part of half a year in conditions that would end a trout in minutes. The trick was worked out by Göran Nielsen's group in Oslo, and the mechanism, published by Catherine Fagernes and colleagues in Scientific Reports in 2017, is about as good an example of evolutionary opportunism as the vertebrates have produced. The problem with running on glycolysis forever is the end product. Lactate accumulates and poisons you, so the carp makes ethanol instead. Somewhere in the ancestry of this group of fish, the whole genome was duplicated, which left spare copies of a lot of enzymes lying around. One of those spares, a component of the pyruvate dehydrogenase complex, drifted into a new job and became a pyruvate decarboxylase, an enzyme otherwise best known from brewer's yeast. Pair that with a specialized alcohol dehydrogenase in the muscle, and you get a fish that converts pyruvate into ethanol. And ethanol is a small, neutral molecule that diffuses straight out across the gills into the pond. So a Crucian carp in a frozen anoxic pond is running fermentation and exhaling the alcohol. The pond, by late winter, is measurably boozy, and the fish, by any instrument you'd care to point at it, is unambiguously alive, while doing something we'd consider incompatible with being an animal. The animal that actually broke the definition, though, is smaller than anything I've mentioned so far, and you may have eaten one. Salmon fishermen and fish processors have known about it for a long time, though not by name. Sometimes you open a fillet, usually a coho or a chinook, and the flesh is studded with small, milky white cysts, like grains of tapioca pressed into the muscle. The industry calls it tapioca disease. It's harmless to people. It's a commercial disaster because nobody buys that fillet. The organism making those cysts is Henneguia salmonicola, and it's an animal. Specifically, it's a cnidarian, from the same phylum as jellyfish and corals, and the Turritopsis we started with. Myxozoans are what happens when a cnidarian becomes a parasite and then keeps simplifying for a few 100 million years. They've lost the tentacles, the nervous system, the gut, the body. What's left is a spore with fewer than 10 cells in it, and the only structure they kept from their jellyfish ancestry is a recognizable stinging capsule, repurposed as an anchor for gripping host tissue. In 2020, a team led by Dorothy Huchon at Tel Aviv University sequenced it, and the result went into the proceedings of the National Academy of Sciences in March of that year. Haenaguia salmonicola has no mitochondrial genome, not a reduced one, not a fragmentary one, none. And when they went looking in the nuclear genome for the machinery that would be needed to replicate and transcribe a mitochondrial genome, most of that was gone too. Under the electron microscope, there are still organelles in there that look like mitochondria, structures with the right general shape, but they're hollowed out. The instructions for running aerobic respiration have been deleted from the animal. Before that paper, aerobic respiration was on the short list of things every animal did. It was considered as fundamental as being multicellular, and a 10-celled parasite, living in the muscle of a salmon, quietly stopped doing it, and nobody noticed until somebody sequenced the thing. Now, the honest part, because this is where the coverage got ahead of the science. We do not know how it makes energy. That's the actual state of knowledge, stated plainly. The paper reports what's missing. It doesn't report what replaced it. The leading guesses are that it steals energy-carrying molecules directly from the salmon's muscle cells, or that it runs some anaerobic pathway nobody has characterized yet. Both are plausible. Neither is demonstrated. There's an animal on this planet whose energy metabolism is an open question. And then there's the case that unsettles me, most precisely because it hasn't been settled. And I want to walk through it properly, rather than tell you the fun version. In the eastern Mediterranean, about 11,500 feet down, there are basins filled with ancient brine. They're called deep hypersaline anoxic basins, and they're properly alien environments. Pools of water so salty it doesn't mix with the seawater above it, forming a sharp boundary you can see on sonar, like a lake at the bottom of the sea, complete with a shoreline. The brine has no oxygen and plenty of sulfide, and it has been that way for thousands of years. One of them is called L'Atalante. In 2010, Roberto da Navarro and colleagues published in BMC Biology that they'd found animals living in the sediments of L'Atalante. Loriciferans, a phylum of microscopic animals that live between sand grains, encased in a sort of armored corset, discovered only in the 1970s. 3 species, new to science. And the claim went well past temporary tolerance. It was that they complete their entire life cycle down there permanently without oxygen. The evidence included staining for living tissue, incorporation of radio-labeled compounds indicating active metabolism, and electron microscopy showing organelles that resembled hydrogenosomes, which are the anaerobic substitutes for mitochondria found in certain single-celled organisms. If that holds, it's the first multicellular animal known to live its whole life without oxygen, and it's a much bigger claim than Haenaguia, because Haenaguia sits inside a well-oxygenated salmon. In 2015, Joan Bernhard and colleagues went back. Sampling those basins is technically brutal, and they couldn't get clean material from the permanently anoxic sediment, so they worked the redox Klein, the transitional layer. They found 10 loriciferans, and they published a challenge, offering alternative readings of the original evidence, including the possibility that the animals were carcasses, preserved rather than living, since anoxic sediments are famously good at preserving things. Da Navarro's group answered that if preservation explained it, you'd expect other small animals to be preserved too. And what they see instead is nematodes and copepods in obvious states of decay in the same sediments, while the loriciferans look intact and stain as living. I've read the exchange more than once, and I don't think either side has closed it. That's the honest position. And it's a good place to notice something. The argument between those two research groups comes down to something much bigger than loriciferans. How you prove an animal is alive when you can't watch it move. Every test they're disputing, the stains, the labeled compounds, the ultrastructure, is a proxy for metabolism. Because metabolism is the definition everyone in the room agreed to use, and the proxies don't agree. So the third definition is in worse shape than the first two. It didn't just get broken by an animal. It turns out you can't always apply it even when you want to. Metabolic rate behaves like a dial rather than a switch, and the ocean has animals sitting at every point along it, down to values that are hard to distinguish from zero with the instruments we have. We'll come back to the bottom of that dial because there are animals waiting down there that finish the argument. First, though, a definition that has nothing to do with chemistry at all. It's the one I suspect you're actually carrying around without having examined it, and it's the one the ocean breaks hardest. Death is the end of everything the animal was. That's the fourth one, and I'd argue it's the definition doing the most quiet work in your head right now because it's a claim about completeness, more than a claim about mechanism. When something dies, the whole package closes. Whatever was going on inside it, all of it, every cell and every process, ends at the same time and for the same reason. Death is total. It takes everything with it. Nobody teaches you that. You absorb it, because it's true of everything you've ever watched die. To break it, go to a mud flat in Maine at low tide with a rake. Soft-shell clams are the ones people dig for steamers along the North Atlantic coast, buried a few inches down, giving themselves away with a squirt of water. Pull one up and open it, and the fluid inside, the hemolymph, which is the closest thing a clam has to blood, should be nearly clear, slightly cloudy at most. In a sick clam, it's milky, thick with cells, so many that the fluid changes color, and when you put a drop under a microscope, the cells are wrong. Round instead of the usual shapes, swollen nuclei, extra sets of chromosomes, dividing at a rate nothing in a healthy clam divides at. They're circulating through the whole animal and pushing into the tissues, crowding out everything that's supposed to be there. It's a cancer. Specifically, it's a leukemia-like condition called disseminated neoplasia. It's been known since the 1970s along the east coast of North America, and it kills the clams. Not occasionally. It has wiped out entire beds, taken commercial fisheries down with it, and it comes through populations in waves. And that last detail is the one that never fit. Cancer is the most private disease there is. That's the whole shape of it. Your own cells acquire mutations, escape the controls that normally keep them in line, and multiply at your expense. And the entire drama is confined to one body. When the body dies, the cancer dies, because it has nowhere else to be. Cancer is a thing that happens inside an individual and is bounded by that individual absolutely. So a cancer that moves through a population in waves is a contradiction. For decades, the explanations were sensible ones. Maybe a virus was spreading and causing the clams to develop their own tumors, the way certain viruses do in other animals. Maybe a pollutant or a toxin was hitting whole beds at once. Maybe these clams shared a genetic susceptibility, and stress in the water was pulling the trigger on all of them together. All three are reasonable. All three are wrong. In April of 2015, Michael Metzger, Carol Reinisch, James Sherry, and Stephen Goff published a paper in Cell that settled it. And the way they settled it is worth walking through because the logic is airtight, and it doesn't depend on believing anybody's interpretation. They had a marker to work with. In the clam genome, there's a mobile genetic element, a retrotransposon, which is a stretch of DNA that copies itself and reinserts the copy somewhere else in the genome. They named this one Steamer. In healthy clam tissue, it sits at a low copy number, a handful of insertions. In the cancer cells, it's amplified enormously, hundreds of copies. The clever part is the locations. Where a retrotransposon lands when it jumps is close to random, which means the specific set of places it has landed in a given cell line is a fingerprint. And the only way two cells share that fingerprint is by descent from a common ancestor cell. Two clams developing their own cancers independently would never produce matching insertion patterns. It would be like two people independently shuffling a deck and getting the same order. The patterns matched. Across clams, across sites hundreds of miles apart. Then they did the test that makes the whole thing undeniable. They took the cancer cells out of a clam, and they took normal tissue from the same clam, and they genotyped both. Microsatellite markers, which are highly variable stretches of repeated DNA used for exactly this kind of identity work, and mitochondrial sequence on top. The cancer cells and the clam did not match. The cancer cells in a clam from Maine matched the cancer cells in a clam from Prince Edward Island. Neither of them matched the animal it was killing, which means the tumor in that clam belonged to somebody else. It arose once, in one soft-shell clam, and that clam died, and its cancer cells got out, and they have been moving from animal to animal ever since. The transmission route is the thing I keep turning over, because it's so ordinary. A clam is a filter feeder. It pumps seawater across its gills all day, straining out anything edible. Cancer cells shed from a dying clam can survive free in seawater for days to weeks, and the next clam downstream draws them in with its food. There's no vector, no clever adaptation for infection, no intermediate host. The animal's method of eating is the route of transmission. It gets sick doing the thing it exists to do, then it got bigger. In 2016, Metzger, working with Antonio Villalba's group in Spain and others, published in Nature. They went looking in other bivalves and found the same phenomenon in mussels, in common cockles, and in golden carpet shell clams. Independent lineages, each one traceable to a single founding animal, which means this hasn't happened once in the history of the ocean. It has happened repeatedly, in separate species, in separate places, and in the golden carpet shells, the genotype came back wrong in a new way. The cancer cells in those animals didn't match golden carpet shells at all. They matched the pullet carpet shell, Venereupus corrugata, a different species living in the same waters. The cancer had originated in one species and crossed into another. And in the region where they sampled, the species it came from wasn't showing the disease. A cancer lineage jumped hosts, established itself in the new species, and left its species of origin behind. 3 years later, in 2019, a team including Marisa Yonemitsu and Metzger published in eLife on the mussels, and the geography of the result is hard to take in. A transmissible cancer lineage that originated in one mussel species, Metallus trossalis, turned up in a different mussel species in South America, and in another different mussel species in Europe. Mussels of that genus don't live in the tropics. There is no continuous band of habitat connecting Chile to the coast of France. The animals themselves could not have carried this across the equator under any circumstances. The explanation the authors point to is us, mussels growing on ship hulls or drawn into ballast water, moved between hemispheres by traffic, a cancer with an itinerary, a cell line that has crossed oceans as a stowaway. The obvious next question is how old the thing is, and in 2023, a genome-wide study of the soft-shell clam lineage, Chenmes Autenses Tulasi Laupoi, from Metzger's group with Samuel Hart as first author, went after it in Nature Cancer. The approach was to find mutational signatures specific to the cancer, measure the rate at which those mutations accumulate using samples collected at different times, and extrapolate backward to the point where the lineage began. The answer is more than 200 years old, with a central estimate somewhere around 420. And I'm going to hold to the standard I set earlier about error bars, because the confidence interval on that number is enormous. The lower bound sits under 200 years, and the upper bound is unbounded, which is a polite way of saying the method can rule out young but can't put a ceiling on old. What the study does establish firmly is centuries. The lineage has also split into two sublineages, one around Prince Edward Island and one in the United States. And their divergence works out to roughly 300 years ago, with an interval running from a bit over a century to 800 years. Take the conservative end of all of that, and you still have a cell line that was already alive and moving between hosts before the United States existed. What that genome looks like now is the part that changed how I think about this. It's a mess. Enormous gains and losses of chromosomal material, structural rearrangements, whole regions where one parental copy has been lost, an expanded mitochondrial genome, transposable elements still jumping around. By every standard of a healthy genome, it's a wreck. And it's viable. It's been a wreck for centuries, and it keeps working. More than that, it's evolving. It has population structure. You can draw a family tree of it, with branches and geography and dates, the way you would for any organism. So put the definition against it. Every single clam that lineage has ever lived in is dead. Not some of them. All of them, without exception. Going back 4 centuries, and the cancer is the reason most of them died. The hosts are a consumable, and the thing doing the consuming has never died at any point in that entire span. Now try to say what it is. It isn't the clam. Its DNA settles that. The animal it's killing is a stranger to it. Is it a separate organism? It has no body, no development, no anatomy, no life cycle, no capacity to exist outside a host for more than a few weeks. Calling it an organism strains the word past breaking. Is it a parasite? Functionally, yes, that's exactly how it behaves, and it's the only word in the vocabulary that fits at all. But it's a parasite, made of the same species it parasitizes, descended from an animal that died before the American Revolution. There's no slot for it. Biology has categories for organisms, for cells, for tissues, for diseases, for lineages, and this thing sits between all of them and refuses to resolve. For our purposes, what matters is simpler. The death of the individual and the death of the lineage have come completely apart. In a clam with this disease, there are 2 distinct entities present with 2 distinct genomes and 2 entirely different fates, and one of them is about to die, and the other one has a future measured in centuries. I should say that this isn't only a marine oddity, because it would be easy to file it that way, and it would be a mistake. Dogs have one. Canine transmissible venereal tumor is a single cell line traced by genome sequencing to one individual dog that lived somewhere in the range of 6 to 11,000 years ago, depending on which estimate you follow, and it's now found on every inhabited continent, having been distributed largely in the last few centuries as dogs traveled with people. Every case in every dog alive today is that same animal's cells. Tasmanian devils have 2. Devil facial tumor disease was first observed in 1996 and has been devastating. A second, entirely independent transmissible cancer was discovered in 2014. Both arose from the same cell type, a Schwann cell from the peripheral nervous system, and they arose separately, in different animals, which is a bleak coincidence for a species already in trouble. 2 in mammals, growing numbers in bivalves. The asymmetry is informative, and the reasons are mechanical. Bivalves are filter feeders, so cells in the water get inside them as a matter of routine. They have open circulatory systems, meaning the hemolymph bathes the tissues directly instead of running through sealed vessels, so a cell that gets in has access to the whole animal. And their immune systems don't perform the kind of tissue-matching rejection that vertebrates perform, in which cells from another individual are recognized as foreign by their surface markers and destroyed. Put those 3 together, and a cancer cell arriving from outside finds an open door, an open road, and no security. We have all 3 of those doors closed, which is why this is vanishingly rare in us, and why the exceptions are so interesting. Which is a good moment to mention the strangest human version of this, though it took a laboratory rather than an ocean to produce it. In 1951, a woman named Henrietta Lacks was treated for cervical cancer at Johns Hopkins, and a sample of her tumor was taken and cultured without her knowledge or consent, which is a fact that deserves stating plainly rather than as a footnote. The cells grew. They kept growing. They became the HeLa line, the first human cells to be maintained indefinitely in culture, and they are still growing today in labs on every continent. And the total mass of HeLa cells produced since then vastly exceeds the mass of the woman they came from. They've changed. Decades of division in flasks have left them with a chromosome count and a genome that no longer matches a human being. Two biologists, Lee Van Valen and Virginia Majorana, argued in the early 90s that HeLa should be classified as a separate species and proposed a name for it. Almost nobody in taxonomy accepted the proposal, and I don't think they were right. What I think is that they were pointing at a real problem with a blunt instrument. And the problem is that our categories have no place to put a human cell line that has outlived its person by 70-odd years and is still evolving. Henrietta Lacks died in 1951. A part of her is measurably alive in more places than she ever visited, and it is no longer, in any strict biological sense, her. That's the same shape as the clam, a cell line whose fate detached from the fate of the individual it came from. So the fourth definition breaks, and it breaks worse than the ones before it. The first three failed because an animal came back from something. This one fails without anything coming back at all. Every host dies exactly the way you'd expect, on schedule, permanently. What survives is a thing we don't have a word for, that carries its own genome and its own history and its own migration record, and that has been continuously alive for longer than most countries. Notice what actually got transferred from one clam to the next. No blueprint was read, no instructions were followed, nothing was rebuilt from a specification. A working cell floated out of a dead animal in a cloud of seawater and got sucked into a living one. And its membranes never opened, and its contents never mixed, and its arrangement was never taken apart at any point in 400 years. Remember that phrase, because everything from here circles back to it. Whether the arrangement can be put back, or in this case, whether it ever had to be. And now let's go back and turn that metabolic dial all the way down, because the animals waiting at the bottom of it break something more fundamental than any of this. In 1959, a Cambridge parasitologist named David Kylan stood up to give the Leeuwenhoek Lecture at the Royal Society and told the room that biology had been working with a false dichotomy. Kylan was not a fringe figure. He'd spent his career on cytochromes, the molecules that carry electrons in the respiratory chain, which makes it a little delicious that he's the man who stood up and argued that some organisms can stop respiring entirely and not be dead. He proposed a name for the condition, cryptobiosis, hidden life, and he defined it as the state of an organism showing no visible signs of life, whose metabolic activity becomes hardly measurable or comes reversibly to a standstill. Read that definition twice. Reversibly to a standstill. He was proposing a third state, sitting between the two we allow for, and he was proposing it with a caution in the wording that survives to this day. Hardly measurable. Kylan knew exactly how much weight that phrase was carrying, and 60-odd years later, it's still carrying it. The best-studied cryptobiote on the planet is an animal you can buy in a packet, brine shrimp, Artemia, the little crustaceans that get sold to children as sea monkeys and to aquarists as fish food, and which live in salt lakes and salt pans in conditions that fluctuate between concentrated brine and dry crust. When conditions turn bad, an Artemia female doesn't release swimming larvae. She releases cysts, embryos arrested partway through development, wrapped in a tough shell, and then dried. Dried is doing a lot of work in that sentence. A dried Artemia cyst contains less than about 7/1000ths of a gram of water per gram of dry mass. There is essentially no free water in it at all. It's not desiccated the way a raisin is desiccated. There is not enough water left in there for the molecules of the cell to be in solution. And John Clegg, working in California through the 1970s, did the careful, unglamorous, definitive work of measuring what metabolism does as you take the water out. He found thresholds. Above a certain hydration, the embryo is developing normally. Below it, development stops, but respiration continues. Below the next threshold, respiration stops too. And below the last one, there is nothing measurable happening in there by any technique he could bring to bear. Those cysts stay viable, dry, for decades. They've been taken down to temperatures approaching absolute zero and brought back. They've been held under vacuum. Commercial cyst stocks are stored dry for years and then hatched on demand, which is an industry, not an experiment. You put them in salt water, and within a day or so, the shell splits and a larva swims out and starts eating. How? Two mechanisms working together, and they're both about what happens to the space that the water used to occupy. The first is chemical substitution. Artemia cysts are loaded with trehalose, a sugar built from two glucose units joined in a way that makes it unusually stable and unreactive. Trehalose has hydroxyl groups arranged so that it can hydrogen bond to the same places on proteins and membranes where water molecules normally sit. As the water leaves, the sugar takes its place, holding the phospholipid heads of a membrane at the right spacing and keeping a folded protein surface satisfied. Without something doing that job, a drying membrane collapses into a different phase and stops being a membrane, and a drying protein unfolds and sticks to its neighbors permanently. The second mechanism is stranger and more important. At those concentrations, the interior of the cell stops behaving like a liquid at all. It vitrifies. It becomes a glass. Glass is an odd material. It has the molecular disorder of a liquid, no crystal structure, no repeating lattice, and the mechanical behavior of a solid. Molecules in a glass are stuck where they are. They can vibrate, but they can't travel. And every chemical reaction in a cell, every single one, requires two molecules to find each other and collide. In a glass, they can't. Diffusion effectively goes to zero. And when diffusion goes to zero, chemistry goes to zero. The cell has been immobilized rather than switched off. Every molecule is still there, in roughly the right place, held in an amorphous solid, waiting. There's a third layer too, and it solves a problem I hadn't thought about until I read the literature. Cells normally maintain their proteins using chaperones, molecular machines that hold things in shape and refold what comes undone. Almost all of them run on the cell's energy currency. In a dried cyst, there is no energy currency being made. So Artemia cysts stockpile two proteins in enormous quantity: a small heat shock protein called P26 and a ferritin relative called artemin. Both are chaperones that work without energy input. The animal packs the shelves before it shuts the doors, with the only tools that function in a building with no power. Now scale that up to an animal with a nervous system, legs, and a brain. Tardigrades, water bears. Everybody's met them by now, and the popular version is exhausting, so let me give you the version that's actually strange. There are marine tardigrades, and one of them, Echinuscoides sigismundi, lives on barnacles in the intertidal zone, which means it dries out and rehydrates with the tide, over and over, as an ordinary feature of its week. When a tardigrade dries, it pulls its legs in and contracts into a barrel shape called a tun, losing the great majority of its body, water. And here the mechanism diverges from the brine shrimp in a way that took the field by surprise. Many tardigrades use little or no trehalose. What they use instead is a set of proteins that are, by the standards of classical biochemistry, defective. Most proteins fold into a specific three-dimensional shape, and their function depends on that shape. These don't. They're intrinsically disordered, floppy in solution, no fixed structure. Two families of them, given the names Cas and SAH in the work from Thomas Boothby and colleagues, are highly expressed during drying. And what they do is form the glass. As water leaves, the disordered proteins gel and then vitrify, creating the same immobilized matrix that trehalose creates in the brine shrimp, by an entirely different route. delete them, and the animals dry badly. Put them into yeast and bacteria, and those cells survive drying better than they should. Same physical solution, different molecules arrived at independently. When you see convergence like that, it usually means the physics left very few doors open. And then there are the two numbers that sent me down this road in the first place. In 2021, a group working on Siberian permafrost published in Current Biology that they had recovered a bdelloid rotifer, a microscopic animal, from frozen ground radiocarbon dated to about 24,000 years ago. They thawed it, it moved, it fed, it reproduced, which for a bdelloid means cloning itself, and the clonal line was still going in the lab. 24,000 years, the last glacial maximum. There were mammoths. 2 years later, in 2023, in PLOS Genetics, another team reported a nematode from Siberian permafrost dated to roughly 46,000 years. It was a species new to science, and they named it Panagrolimus kolymaiensis after the Kolyma River. They sequenced its genome and found that the machinery it uses overlaps substantially with what the laboratory nematode C. elegans deploys in its dour larva, the survival stage it forms, under stress, including the trehalose pathway, which makes it the ordinary survival toolkit of a very ordinary worm left running for 46,000 years. I want to be square with you about what those results are and aren't, because they're the kind of finding that gets repeated carelessly. They are individual survivors. Both teams processed a lot of material to get what they got, and the vast majority of what's in ancient permafrost is not revivable. Both teams took contamination seriously, because a modern rotifer falling into a sample would produce exactly this result and be a catastrophe for the paper, and both used sterile technique and dated the surrounding material rather than the animal. The dates are for the sediment, which is the standard approach and comes with the standard assumption that the animal was deposited with it. I think both results are probably sound. I also think anyone who tells you these animals were in perfect suspended animation is skipping something, because the dry or frozen state is not perfectly stable. Damage accumulates in there. Background radiation breaks DNA, whether or not the organism is doing anything about it, and slow chemical processes nick and cross-link molecules over time. Rotifers and tardigrades come out of long dormancy with fragmented genomes, and a large part of what they do in the first hours after rehydration is emergency DNA repair. That's why the ancient survivors are rare. The state isn't timeless, it just decays extremely slowly, and the odds of any individual making it through get worse and worse as the millennia stack up. Rehydration is dangerous too. Bring a dried tardigrade back too fast, and it dies, because the glass has to soften in an orderly way, and the membranes have to reform before the machinery restarts. But look at what we've done to the third definition. Death is the cessation of metabolism. Take that seriously, apply it honestly, and a dried Artemia cyst is dead. Not dormant, not slowed, dead by the criterion, with no measurable chemistry occurring anywhere inside it. A rotifer in permafrost at year 20,000 is dead by that criterion, and stays dead for another 4,000 years, and then reproduces. You can try to rescue the definition by saying the metabolism is still going, just too slowly to detect. People do say that, and it might even be true. Though when I read the arguments carefully, what I mostly see is a disagreement about instruments rather than about organisms. If your criterion for being alive is activity below the threshold of every technique we own, your criterion has stopped being about biology and started being about laboratory equipment. The other option is to accept what the brine shrimp is telling us, and it's a very large idea. Being alive might not be an activity at all. An arrangement of molecules can host a process, and when conditions allow, the process runs. When conditions don't, the process stops and the arrangement persists. The dried cyst is the arrangement without the process. What makes it different from a dead cyst, and a dead one looks identical from the outside, is that the arrangement is intact and can start again, which sends us straight back to the phrase I flagged earlier. Whether the arrangement can be put back, we'll need it again. For now, let's go after the definition most people actually mean when they use the word death in ordinary conversation, because it's the one where the ocean has both the best data and the loudest arguments. Death is what aging arrives at. That's the definition doing most of the work in ordinary human life, and it's so deeply built in that we hardly recognize it as a claim. There's a span. The span has a length. The length is a property of the kind of animal you are, roughly fixed, with some variation for luck and habits, and at the end of it the body has worn out and stops. Mice get 3 years. Dogs get about 15. We get 80 or so. Everything runs down. To test this one properly, I need to be precise about what aging actually means, because the everyday sense of the word and the technical sense come apart. And the gap between them is where all the interesting animals live. In 1825, a British actuary named Benjamin Gompertz, working for an insurance company and trying to price annuities correctly, noticed something in mortality tables. The chance of a person dying in the next year isn't constant. After early adulthood, it rises, and it rises in a very particular way. It doubles at a steady interval. For adult humans, that interval sits somewhere around 7 or 8 years. Your risk of dying at 40 is roughly double your risk at 32, and your risk at 70 is enormously higher than at 40, and the curve doesn't bend down, ever. That doubling is what aging is technically. Senescence is a rising mortality rate, plus declining physical function, plus declining reproductive output, which means something counterintuitive. A population where 1% of individuals die every year, forever, with the rate never changing, isn't aging at all. Every individual in that population will eventually die. The average lifespan is 100 years, and yet no individual is deteriorating because a 1,000-year-old member of that population faces exactly the same risk this year as a 10-year-old. Death happens. Aging doesn't. Caleb Finch at the University of Southern California gave that condition a name in 1990, negligible senescence. No detectable rise in mortality rate with age, no decline in reproduction, no measurable physiological deterioration. He listed it as a possibility, a corner of the space, and the question was whether anything actually lived there. The ocean is full of things living there. Start with the fish that made the aging field pay attention, and I'll say at the outset that its main claim to fame is the range within its own family rather than the record itself. Rockfish. Genus Sebastes. Somewhere north of 100 species, most of them on the Pacific coast of North America. And if you've eaten fish and chips on the West Coast, you've probably eaten one. They're the spiny, blotchy, unhurried fish you see hanging motionless over reefs and rock piles in colors that run from drab brown to a red, so intense it looks artificial. The rough-eye rockfish, Sebastes aleutianus, lives in deep water in the North Pacific, off Alaska and British Columbia, and the maximum documented age for the species is over 205 years. 205. A rough-eye rockfish hauled up off Alaska in the last decade could have hatched while Thomas Jefferson was president and spent the entire 19th and 20th centuries drifting over the same stretch of rock, eating shrimp. That number by itself is remarkable. What makes it scientifically important is the company it keeps. Sebastes minor, in the same genus, closely related, similar body plan, similar ecology in broad terms, lives about 11 years. 11 and 205. Same genus, which tells you that lifespan behaves like a setting rather than a deep property of being a fish, or a vertebrate, or an animal. Evolution can turn that dial through a factor of about 20 within a single group of closely related species, which means whatever aging is, it's under fairly direct genetic control, and it responds to selection like any other trait. Now, before we go further, I owe you the methods, because a number like 205 years is worth exactly as much as the technique that produced it. Fish are aged from otoliths, ear stones. Every bony fish has 3 pairs of them. Small calcium carbonate structures in the inner ear used for balance and hearing, and they grow by accretion, adding layers on the outside throughout life. Those layers vary with season, so a sectioned otolith under a microscope shows alternating bands, and you count them like tree rings. For a 5-year-old cod, this is routine. For a 200-year-old rockfish, it is not. And here's the problem. Growth slows dramatically with age, so the bands from the last century of the animal's life are crammed into the outer edge of the stone, thinner and thinner, until you're squinting at something at the limit of what a microscope resolves. Counting errors in that region are not just possible, they're expected. And different readers looking at the same otolith will not always agree. So the field validated the counts a different way using physics. Otoliths incorporate a little radium as they form, and radium-226 decays to lead-210 with a known half-life. In the core of the otolith, the material laid down when the fish was young, the ratio of those 2 isotopes has been quietly running as a clock ever since, indifferent to how many bands anyone can resolve. Measure the ratio, do the arithmetic, and you get an age estimate that owes nothing to counting rings. When that was done on rockfish, the isotope ages agreed with the band counts. 2 independent methods, based on entirely different physics, converging on the same extreme numbers. That's what makes the 200-year figure something I'm comfortable repeating. And it's the standard I'd like to apply to every age claim in this section, including the famous one. Living a long time is only half of what makes rockfish interesting. The other half is the condition they're in near the end of it. An 80-year-old rough-eye rockfish is still growing slowly. Its tissues don't show the accumulated damage you'd expect. And its reproductive output goes in the direction opposite to ours. In us, and in most animals we know well, reproduction declines with age in both quantity and quality. In rockfish, it improves. Larger, older females carry vastly more eggs because egg number scales steeply with body size, and the larvae they produce are measurably better. Work on black rockfish by Stephen Berkeley and colleagues published in Ecology in 2004 found that larvae from older mothers grew faster and survived starvation for substantially longer than larvae from young mothers, apparently because older females provision their eggs with bigger oil reserves. Fisheries scientists have a nickname for these animals, and I love that it made it into the literature: big, old, fat, fecund female fish. The consequence is uncomfortable and worth stating plainly. Fisheries that target the largest individuals are removing the animals that contribute most of the reproductive success of the population, and doing it at a rate that a 200-year life history was never built to absorb. A species that takes 50 years to reach its most productive stage does not recover on the timescale of a management plan. In 2021, a team at Berkeley with Sri Rohit Raj Kalora and Gregory Owens as co-first authors published a genomic assault on the question in science. They sequenced 88 rockfish species with high-quality long-read genomes for 6 of them, and asked what changes track with lifespan across the group. They found around 137 genes whose patterns line up with the differences in lifespan, and the categories they fall into are the ones you'd bet on: DNA repair, insulin signaling and nutrient sensing, and immune regulation. Lifespan also tracks with body size and with depth, which makes ecological sense, since a big fish in deep cold water has a slower metabolism and fewer things capable of eating it. Once again, and I'll keep flagging this because it matters, that's a comparative genomic study. It identifies candidates by correlation across species. It's a map of where to dig. Nobody has taken a short-lived rockfish, edited those genes, and produced a long-lived one. And until somebody does, the causal claim is unfinished. Fish, though, are amateurs at this. Arctica islandica is a clam, the ocean quahog. It lives buried in sand and mud across the North Atlantic. From New England to Iceland to the Irish Sea, it's about the size of a fist, and it is fished commercially in enormous quantities for chowder. There is nothing about it that looks like a scientific celebrity. Its shell grows in annual increments, the same way a tree adds a ring, and the increments are sensitive to conditions in the water that year, which means quahog shells are climate records. Researchers, particularly the group at Bangor University in Wales, developed the technique of cross-dating them, matching the pattern of wide and narrow years between individuals, exactly the way tree ring scientists match timbers. Overlap enough shells, including dead ones from the seafloor, and you can build a continuous, annually resolved record of North Atlantic conditions stretching back more than 1,000 years, with each year identified precisely. Think about what that requires. It requires individual animals, these that live for centuries, routinely, in numbers large enough to sample. In 2006, a Bangor team dredging off the north coast of Iceland for exactly that purpose brought up a haul of quahogs, froze them for later analysis, and got on with the work. One of the shells in that batch was counted in 2007 and came out at 405 to 410 years old, which was already a record for any animal. In 2013, they went back and did it properly. The original count had been taken from bands on the outside of the shell and in an animal that old the late life increments on the outer surface are compressed to the point where several years merge into what looks like one. So the team sectioned the hinge, where the increments stay better separated, counted there, cross-dated the pattern against their master chronology, and checked the result with radiocarbon. 507 years. The clam was given the nickname Ming because it settled onto the seafloor around the year 1499 during the Ming Dynasty, 7 years after Columbus crossed the Atlantic and well before Shakespeare was born. It sat in the mud off Iceland, filtering, laying down one ring a year, through the Reformation, the Little Ice Age, the entire Industrial Revolution and both World Wars, and it was still doing it when the dredge came down. And yes, the analysis killed it, and yes, the press had a field day with that. I think the outrage was aimed at the wrong target. Nobody knew what they had. It was one clam among many in a routine sample. Its age was determined after the fact. And ocean quahogs are harvested commercially by the ton every year with no headlines at all. If a 500-year-old animal being killed is upsetting, then the commercial fleet is where the story actually is, and it has been running for decades. What actually matters is what the quahogs revealed about aging. Their mortality rate does not measurably climb with age in the older part of the population. A 300-year-old quahog faces roughly the risk a 100-year-old faces. The physiology backs it up. These animals have very low metabolic rates. They tolerate long stretches without oxygen when they're burrowed in. And comparative work has found their tissues unusually resistant to oxidative damage, partly because of the composition of their cell membranes. Membranes rich in highly unsaturated fats are chemically vulnerable. They oxidize readily, and the damage propagates. And long-lived mollusks tend to build membranes with less of that vulnerability. Cheaper to maintain, harder to damage. They still die, something eats them, or a dredge takes them, or a bad year in the sediment takes them. What they don't do is fall apart on a schedule, which leaves one very obvious question hanging over everything I just said. And it's the question I want to spend real time on, because it's where this subject gets properly difficult. How do you know how old an animal is when nobody was there when it hatched? There's an animal in the North Atlantic that made the field confront that question directly, because with this one, every standard technique failed. The Greenland shark is a large, slow, cold water shark that can reach something over 20 feet. It lives across the Arctic and the northern North Atlantic, from under the sea ice down to depths of well over a mile, in water that hovers around freezing. It cruises at roughly half a mile an hour, which is a walking pace so leisurely that biologists spent years wondering how it catches anything at all, and its flesh is loaded with urea and a compound called trimethylamine oxide that make it inedible fresh. Icelanders deal with that by burying and drying it for months to make hakarl, a food that people either revere or use as a dare, and most of them are blind. A parasitic coat pod called Omatocoida elongata attaches to the cornea and hangs off the eye like a small pale streamer, and it's found on a large majority of the Greenland sharks anyone examines. The sharks appear entirely untroubled by this, which tells you roughly how much they were relying on vision in the first place, a mile down in the Arctic dark. Now, how old is one? A bony fish gives you otoliths. A shark doesn't have any. Sharks have cartilage skeletons, and for many species you can section a vertebra and count growth bands in the calcified rings, which works reasonably well for, say, a mako. Greenland shark vertebrae are poorly calcified and don't lay down bands you can read. There are no scales to count, no shell, no annual increments anywhere in the animal. So Julius Nielsen and his colleagues went after the one tissue in a vertebrate body that never changes, the middle of the eye. The lens of an eye is built from proteins called crystallins, packed at extraordinary density and arranged in layers to be transparent. The layers at the very center, the lens nucleus, are laid down during embryonic development, before the animal is born, and then they're done. There's no blood supply in there, no turnover, no replacement. The proteins in the nucleus of your own eye lenses were assembled before you were born, and they are still the same molecules today. It's the closest thing a vertebrate has to a sealed sample of itself at the moment of formation, which makes it a place to look for a date stamp. And the 20th century obligingly provided one. Atmospheric nuclear weapons testing through the 1950s and early 60s roughly doubled the amount of radiocarbon in the atmosphere. Then, the Partial Test Ban Treaty in 1963 stopped atmospheric testing, and the excess has been draining out of the air ever since, absorbed into oceans and plants. The result is a sharp global spike in the carbon record with a known shape and known dates, and geochemists call it the bomb pulse. Anything that incorporated carbon during those years carries the signature. Anything that formed before doesn't. Nielsen's team, publishing in Science in 2016, obtained eye lenses from 28 female Greenland sharks that had come up as bycatch in Greenland surveys, dissected out the nuclei, and ran radiocarbon on them. The two smallest sharks carried the bomb signal, meaning they'd formed after the early 1960s. Everything larger was pre-bomb. Those two small animals became the calibration points, and from there the team built a growth model relating body length to age and applied it to the rest. The largest shark in the sample, the Warsinsan, was just over 16.5 feet long, and the model put its age at 392 years, plus or minus 120. I want to stay on that second number for a minute because it's the part that vanishes in every retelling, and it's the most honest thing in the paper. The uncertainty on that estimate is 120 years in each direction. The defensible range for that individual runs from about 272 years to about 512. The authors were careful about it. The claim they actually made in the paper is that the Greenland shark's lifespan is at least 272 years, because that's the number the evidence supports at the low end. And they let the headline writers do the rest. Where does that uncertainty come from? Several places stacked. Radiocarbon in the ocean isn't the same as radiocarbon in the air. The surface ocean takes up atmospheric carbon with a delay, and deeper water carries carbon that hasn't seen the surface in centuries. So converting a marine radiocarbon measurement into a calendar date requires a reservoir correction, and that correction is itself an estimate with its own error. Then there's the model. Only the two smallest sharks were dated directly against the bomb pulse. Every other age in the study comes from a growth curve fitted to those anchor points, and extrapolating a growth relationship from a couple of juveniles out to a 16-foot adult is exactly the kind of extension where small errors in the curve become large errors in the answer. And there were 28 animals, all female, all obtained as bycatch, which is not a random sample of the population, none of which makes the result wrong. I think the result is very probably right in its broad shape, and the Greenland shark is almost certainly the longest-lived vertebrate we know of, by a wide margin. But 392 is a point estimate sitting in the middle of a wide distribution, and speaking it without its error, Barr turns a careful piece of science into a trivia fact. The same paper produced a second number that I find harder to shake than the first. Female Greenland sharks appear to reach sexual maturity at around 13 feet, and the growth model puts that length at roughly 150 years of age, with all the same uncertainty attached. 150 years to first reproduction, which means a female Greenland shark taken as bycatch in a trawl today might be an animal that hatched around the time of the American Civil War and had not yet reproduced. There is no fisheries management framework on Earth designed for that. Our entire regulatory vocabulary, spawning stock, recovery time, sustainable yield, was built around animals that turn over in years. Let me widen this out because the Greenland shark is the extreme case of a problem that runs through every long-lived animal I've mentioned. Every method for aging an animal you didn't watch grow up is a clock. And every clock needs to be set against something. Counting growth increments works when the increments are annual, resolvable, and validated. It gets unreliable in old animals, when growth slows and the bands compress, which is exactly the population you care about. Lead radium dating works because radioactive decay ignores biology, but it assumes the tissue took up radium once, early, and never exchanged any with the surroundings afterward. Bomb radiocarbon works beautifully, and only for animals whose lives straddle the 1960s, and only in tissues that are metabolically inert. There's another approach used on marine mammals that's worth knowing about because of how it was checked. Amino acids come in two mirror image forms, and living organisms build proteins almost entirely from one of them. In inert tissue, over long periods, they slowly flip to the other form at a temperature-dependent rate. Measure the ratio in the eye lens of a whale, and you get an age. Applied to bowhead whales in the late 1990s, that method returned ages over 200 years, with one individual estimated near 211. That should have been treated with heavy skepticism, and it was. And then the check arrived from an unexpected direction. Whalers in Alaska recovered stone and ivory harpoon points of a style that went out of use in the late 1800s. Embedded in the blubber of bowheads taken in the modern era. A physical object with a manufacturing date, still inside a living animal. Two completely unrelated lines of evidence, one chemical and one archaeological, pointing at the same conclusion. That's what a validated age claim looks like, and it's rare. Right now there's a lot of interest in epigenetic clocks, which read chemical marks on DNA, methyl groups attached at specific sites, that change with age in patterns regular enough to predict it. In humans and mice, these are remarkably accurate. Extending them to other species is an active field, and they run headlong into a beautiful catch. To calibrate a clock, you need animals of known age. For a Greenland shark, there are none. There has never been a Greenland shark of known age, because nobody has ever watched one from birth, and nobody currently alive will. Any clock built for this animal has to be calibrated against ages that were themselves estimated by another method whose own calibration is uncertain. You can do useful work under those conditions, and people are, but you can't escape the fact that the foundation is an estimate. So, does the Greenland shark break the definition of death as the end of a lifespan? Honestly, no. It stretches it. A Greenland shark almost certainly does age. It just does everything at a rate set by water near the freezing point, and a body running at a metabolic idle. And when every biological process is that slow, 3 centuries is the natural result rather than a miracle. The quahog and the rockfish break it. Because what those animals demonstrate is that the mortality curve doesn't have to rise. And once you accept that, the idea of a lifespan comes apart in your hands. We talk as though every animal has a countdown running inside it, an allotment being spent. There's no such thing. What exists is a probability of dying in the next interval, and in most animals we're familiar with, that probability climbs steeply with age, so the allotment feels real. Gompertz put a number on it. For humans, doubling every 7 or 8 years. That doubling is the origin of the entire human intuition about mortality. It's why age feels like a countdown. It's why a birthday means something. In an animal whose curve is flat, none of that exists. A 300-year-old quahog isn't nearer the end than a 50-year-old quahog. There is no end to be nearer to. There's just a level chance of something going wrong this year. The same level chance it faced last year. And it will keep facing that same chance until one year the coin lands the other way. So the 6th definition of death describes a curve, a particular shape of mortality risk over time, which our species happens to have, and which we mistook for a feature of being alive. 6 definitions down. Every one of them was really a description of us, which leaves the last place to look, and it's the one I've been saving because it's the only definition on the list that isn't about organisms at all. Death is what happens to cells. That's the 7th definition, and on the face of it, it's the most rigorous of the lot. Life is cellular. Every living thing on this planet is made of cells, or is a cell. So whatever death is, it must ultimately be something that happens to cells. And if we can characterize that properly, then all the confusion at the level of jellyfish and clams is just noise on top of a clean underlying process. For a long time, everyone assumed the cellular process was as simple as it sounds. A cell is a bag of chemistry. Damage it enough, starve it of energy, poison it, and it stops working and comes apart. Cell death was a synonym for cell failure. Then people started looking at dying cells properly under a microscope, and it turned out there were 2 of them. In 1972, 3 pathologists, John Kerr, Andrew Willey, and Alistair Currie, published a paper in the British Journal of Cancer describing a form of cell death that looked nothing like the one everybody knew. They gave it a name borrowed from Greek for the falling of leaves from a tree in autumn. Apoptosis? Set the 2 side by side because the contrast is the whole story. A cell dying of necrosis, the ordinary kind, swells. Its energy supply fails, which means the pumps in its membrane that maintain the balance of ions stop running, which means sodium and water flood in and keep flooding in. The organelles swell. The membrane stretches and finally ruptures, and everything inside the cell, enzymes, ions, proteins, the whole toolkit of a working cell spills out into the surrounding tissue where none of it belongs. The neighbors get chemically assaulted. The immune system reads that spill as an alarm, arrives in force, and inflammation follows. That's a burst pipe. A cell undergoing apoptosis shrinks. It pulls away from its neighbors. The chromatin in the nucleus, normally spread through the nuclear space, condenses into dense crescents pressed against the inside of the nuclear envelope. The cytoskeleton gets taken apart in an orderly sequence. The surface of the cell erupts into small bubbles, a process called blebbing, and then the whole thing partitions itself into membrane-wrapped packages, each one sealed, each one containing a portion of the cell's contents with nothing leaking out. Those packages are eaten by neighboring cells and by macrophages, digested and recycled. Nothing spills, nothing inflames. In a tissue where apoptosis is happening, you can look and see almost nothing because the cleanup is fast and the evidence gets swallowed. And there's a specific chemical signal that makes the cleanup work, which I find quietly moving in a way I can't fully justify. Cell membranes are built of 2 layers of phospholipids, and the composition of the 2 layers is deliberately kept different. One lipid in particular, phosphatidylserine, is normally held on the interface by an enzyme that constantly flips it back inward, running on the cell's energy supply. When a cell commits to apoptosis, that arrangement is deliberately reversed, and phosphatidylserine appears on the outside. Immune cells read that as a signal. The literature calls it the eat me signal. The dying cell is displaying on its own surface a request to be consumed. So, 2 ways to die, one of them a catastrophe and one of them a procedure, which raised the obvious question, and answering it produced one of the great pieces of 20th century biology. In the 1960s, Sidney Brenner went looking for an animal simple enough to understand completely. He settled on Caenorhabditis elegans, a soil nematode about a millimeter long, transparent, that reproduces mostly as a self-fertilizing hermaphrodite and grows from egg to adult in about 3 days. John Sulston then did something that still sounds unreasonable. He sat at a microscope and traced the entire cell lineage of the animal. Every division, from the fertilized egg to the adult, watching cells split and migrate and differentiate for years, and wrote down the family tree of every cell in the organism. Here's what came out of it. During development, the hermaphrodite generates 1,090 somatic cells. The adult has 959. 131 cells are made and then destroyed. And it's the same 131 every time. In every individual worm of that species, the same identified cells, in the same order, at the same points in development, are produced and then eliminated. Sulston could name them. The lineage chart has the deaths marked on it, like any other developmental event, because that's exactly what they are. Think about what that means. Death at the cellular level is scheduled. It's in the blueprint. Building this animal requires making 131 cells whose entire purpose is to be made and then removed. And the removal is as precisely specified as the construction. Robert Horvitz then went after the genes responsible and found them by looking for mutant worms in which the deaths failed to happen. He identified 2 genes, SED3 and SED4, both required for the deaths to occur, and a third, SED9, which does the opposite and protects cells from dying. Brenner, Sulston, and Horvitz shared the Nobel Prize in 2002, awarded for the genetic regulation of organ development and programmed cell death, which is a strange thing to win a prize for and completely deserved. The reason it mattered so much beyond worms is what happened when people looked for the equivalents in us. They're all there. SED3 corresponds to a family of enzymes we call caspases, protein-cutting enzymes that sit dormant in every one of your cells waiting to be activated. SED4 corresponds to a protein called APAF1, and SED9 corresponds to a gene called BCL2, which is where the story takes a turn because BCL2 had already been discovered in cancer research as a gene that goes wrong in a form of lymphoma. Follow that. A gene that causes cancer, and what it does is prevent cells from dying. That reframed a whole discipline. Cancer had been understood as a disease of cells dividing too much. BCL2 showed that a tumor can also arise from cells failing to die on time. Every day your body produces cells with damaged DNA, And a large part of your defense is that damaged cells detect their own damage and initiate their own destruction. Break that. And the cells don't have to divide faster to accumulate. They just have to stop leaving. The machinery itself, briefly, because it's worth seeing how deliberate it is. When a cell commits, its mitochondria release cytochrome C into the cytoplasm. Cytochrome C is a component of the respiratory chain, one of the molecules Keelan spent his career on. And in the cytoplasm, it has a completely different function. It binds APAF1, and 7 of those complexes assemble into a wheel-shaped structure called the apoptosis. The apoptosis activates an initiator caspase. That caspase activates executioner caspases, and those go to work on hundreds of specific targets throughout the cell, cutting each at a precise site. One of the targets is a protein that holds a DNA-cutting enzyme in check. Cut it, and the nuclease is released and chops the genome between the spools that DNA is wound around, producing fragments in regular-sized steps. There's no damage anywhere in that sequence. It's a program running to completion, using enzymes the cell built and maintained for exactly this occasion, and the cell pays for it with its own energy right up to the end. Apoptosis costs the cell energy. A cell too depleted to run the program dies the messy way instead. Now, how much of this is happening? Estimates of how many cells undergo apoptosis in an adult human body run into the tens of billions per day. Those figures are rough, derived from turnover rates in tissues like gut lining, skin, and blood. And different methods give different answers, so treat them as an order of magnitude rather than a measurement. But even the low end of the range describes a situation people don't usually picture. A body in which an enormous population of cells is destroyed and replaced continuously, forever, as the ordinary condition of being healthy. This shaped you. Your hands were webbed as an embryo, and the tissue between the developing fingers was removed by programmed cell death. The immune system you're running was assembled by generating a vast library of immune cells with randomly generated receptors and then killing off, in the thymus, the ones that recognized your own tissues. Roughly speaking, most of the T cells you ever make are destroyed before they leave. Your tolerance of your own body is built on a very large amount of intentional cell death. And in the ocean, this shows up in forms that are almost theatrical. Take a sea squirt. Ciona intestinalis begins as a swimming larva shaped like a tadpole, with a muscular tail stiffened by a notochord and a dorsal nerve cord, which is the architecture that defines our entire phylum. The larva swims for a matter of hours, finds a hard surface, and glues itself down head first. Then it resorbs its own tail. The tail regression is driven by caspases. Block caspase activity in the tail doesn't retract properly. An animal on our own branch of the tree, having built the defining structure of the chordates, deletes it on a schedule, using the same enzymes that dispose of your worn-out cells, and settles down to spend the rest of its life as a sessile filter feeder. The same thing runs through echinoderm metamorphosis, where the elaborate swimming larva of a sea star or sea urchin is largely dismantled by programmed cell death as the juvenile forms within it. And it runs through the sea cucumber story from earlier. Work mapping the spatial dynamics of regeneration after evisceration finds coordinated waves of cell death and cell proliferation moving through the tissue, with the destruction as carefully placed as the building. Then, sometime in the last 20 years, the tidy picture from 1972 fell apart. It turned out necrosis isn't always an accident either. There's a form called necroptosis, which proceeds through a signaling cascade involving a pair of dedicated kinase proteins, ending with a protein called MLKL that travels to the cell membrane and punches holes in it. The result looks exactly like classic necrosis. The cell swells, the membrane fails, the contents spill. The immune system responds to the mess, and every step of it is genetically programmed, with dedicated proteins that exist for no other reason. There's ferroptosis, described by Scott Dixon, Brent Stockwell, and colleagues in 2012, in which the cell dies from iron-dependent oxidation of the fats in its membranes, a chain reaction that spreads through the lipid layer. It has its own dedicated brake, an enzyme called GPX4, which normally suppresses it continuously. Remove the brake, and the cell dies this specific way. There's pyroptosis, which involves proteins called gasdermins that assemble into pores in the membrane, used deliberately by the immune system against infected cells, so the clean binary is gone. What we have now is a spectrum of at least a dozen named modes of cell death, several of which are programs disguised as accidents, and the boundaries between them keep getting redrawn. And that leaves the seventh definition in a stranger position than any of the others. The other six failed because some animal walked through them. This one fails from the inside. Death at the cellular level turns out to be, in a large fraction of cases, something the organism does on purpose, using dedicated machinery, at a scale of billions of events a day, as a requirement of staying alive. You can't define the death of an animal in terms of the death of its cells, because its cells are dying constantly, and that's the healthy state. A tissue holding steady is a place where cell birth and cell death are balanced against each other, and the balance is the thing that's alive. Which sets up the last question I have, and this one actually goes somewhere. If cell death is a program, can the program be switched off? The answer is yes, and the animals that do it are the ones we've already met. Go back to the mussel clamped shut on the shore, and the carp under the ice, and the squid turning itself down for the day. Every one of those animals has a problem I skipped over at the time, and it's a problem that only becomes visible once you know how cell death is wired. The triggers for apoptosis include exactly the conditions those animals impose on themselves. A cell that runs low on energy, that accumulates damaged proteins, that detects its mitochondria in trouble, is a cell whose internal sensors are screaming that something is badly wrong. And the response those sensors are built to produce is self-destruction. That's the point of the system. A cell in that much trouble is a liability, and the tidy thing to do is dismantle it and let a neighbor take over. Now imagine that state persisting for 4 months. An animal that goes anoxic all winter, if it left its normal cellular programming running, would emerge in spring having deleted an enormous fraction of its own tissue. The machinery would be doing exactly what it was designed to do, in a situation it was never designed for, and the result would be catastrophic. So the anoxia-tolerant animals hold it down. Comparative work across anoxia-tolerant vertebrates, a lot of it from Kenneth Story's group at Carleton University in Ottawa, has repeatedly found the same signature during long anoxic bouts. The pro-death machinery suppressed, the protective side of the balance raised. Caspus activity staying low through periods that ought to trigger it everywhere. The schedule is put on hold, deliberately, for the duration. And the way it's held is worth understanding because it's the mechanism that makes the whole strategy of suspension possible. You could imagine shutting a body down by changing which genes are expressed. Turn off the genes for the expensive processes, wait, turn them back on. The problem is that gene expression is slow and expensive. Making new proteins is one of the largest energy costs a cell has, and an animal that's shutting down to save energy can't afford to spend a fortune on the shutdown. What they use instead is chemistry that's already in place. Reversible phosphorylation. A phosphate group gets attached to an enzyme at a specific site, and the enzyme's shape changes, and it stops working. Remove the phosphate, and it starts again. It's fast, it costs almost nothing, it's applied by enzymes that already exist in the cell, and it can be coordinated across dozens of pathways at once. A whole animal can be turned down by a few % of its normal rate, through the addition of phosphate groups to the right set of proteins, and turned back up the same way. There's a second piece proposed by Peter Hochachka and Science in 1986, and it addresses the single biggest running cost of a resting cell. That cost is holding ion gradients across membranes. Cells maintain a constant difference in sodium and potassium between inside and outside, and they do it by pumping continuously against a continuous leak. And in a resting animal, that pumping is one of the largest items on the energy bill. Hochachka's argument, which came to be called channel arrest, was that anoxia-tolerant animals reduce the leak, closing or down-regulating the channels through which ions escape, so that the pumps have far less to do. Lower the leak, lower the bill. Between those two ideas, you have most of the machinery of biological suspension. Turn the enzymes off with phosphate. Seal the membranes so the gradients hold. Hold down the self-destruct programs, so nothing dismantles itself while it waits. And then the animal comes back, and everything is where it was left. There's a version of this that goes further, and it takes us back to where we started, which I'll do once and then leave alone. When a Turritopsis medusa collapses, its cells are experiencing precisely the stress that normally initiates programmed death. Starvation, injury, structural failure of the tissue. Some of them do die. The reversal isn't a bloodless process, but a substantial part of that population does something the textbook doesn't have a slot for. Instead of executing the program, the cells shut down their specialized identity and adopt a different one. The workforce gets retrained instead of dismissed, which puts a strange light on that whole phenomenon. The remarkable part of reverse development might have less to do with growing new tissue than with the number of cells that decline to die when they're told to. There's one more twist here, and it's what turned this whole subject around for me. In a lot of regenerating systems, cell death actively causes regrowth. The phenomenon is called apoptosis-induced proliferation, and it's been documented across a range of animals. Cells undergoing apoptosis release signaling molecules on their way out, and those signals reach neighboring stem cells and instruct them to divide. Block the cell death, and the regeneration slows or fails. The dying cells are issuing the order to rebuild. Look at the shape of that for a second. At the cellular level, death is a construction tool. It's how a hand gets fingers. It's how a sea squirt loses a tail it doesn't need. It's how a body knows where to grow. Death at that scale is one of the moves that living things make. So let's take stock, because we've now run 7 definitions through the ocean, and I want to lay them out. Death as the end of a one-way life cycle, broken by a jellyfish that runs its life cycle backwards using cells that change job. Death as the destruction of the body, broken by a sea star arm that walks away and becomes a sea star, by an animal that throws out its own organs and regrows them in a fortnight, by a colonial chordate that rebuilds a complete body out of its own plumbing. Death as the cessation of metabolism, broken by a 10-celled parasite that deleted the instructions for aerobic respiration and is still here, and challenged unresolved by animals in a brine pool at the bottom of the Mediterranean. Death as the end of everything the animal was, broken by a cell line that has outlived every clam it ever occupied for centuries, and crossed between species and between hemispheres, while every one of its hosts died on schedule. Death as metabolism reaching zero, broken by a brine shrimp cyst with no measurable chemistry occurring in it, by a rotifer that spent 24,000 years in permafrost, by a nematode that spent 46,000. Death as the end of a lifespan, broken by clams and rockfish whose risk of dying doesn't climb with age, which reveals that the lifespan we imagine as a countdown is the shape of a curve, and the curve is a variable. Death as the death of cells, broken from the inside by the discovery that a healthy body destroys billions of its own cells a day on a schedule, using dedicated equipment as a requirement of being healthy. 7 definitions, 7 failures. And I'd like to point out something about the pattern of the failures, because it isn't random. Every single one of those definitions was a description of us. A one-way life cycle is our life cycle. A body that fails as a unit is our body, centralized and non-redundant. A metabolism that can't be interrupted is our metabolism, with a brain that burns a fifth of the budget and holds no reserve. Cells with nowhere to go once the body stops are our cells. Metabolism that must never stop is our constraint. A rising mortality curve is our curve, and Gompertz found it in insurance tables. And cells that die only when something goes wrong was our assumption, right up until somebody counted them properly in a worm. We took the specific way that a large, warm, centralized, fast metabolizing, steeply senescent land vertebrate stops working, and we called it death. And then we went looking for animals that break the rules. They're not breaking rules. They never agreed to them. So the question that's left is whether anything survives. Whether there's a line that holds against a jellyfish and a rotifer and a 500-year-old clam, or whether the whole concept dissolves into a family of local descriptions with nothing in common. I think something survives. I think it's a real line, and a sharp one, and it's much smaller than the one we've been using. Here's how to find it. Go back through every case in this script where something came back, and ask what was true in all of them. The medusa that collapsed into a blob. Its cells were intact the whole time. Membranes unbroken, genome complete, proteins folded. What changed was the configuration, and the configuration was rebuilt from parts that were all still there. The sea star arm crawling across the reef with no mouth. A complete working piece of tissue carrying stem cells and the positional information needed to work out which end is which and what's missing. The muscle clamped shut for weeks against a dead tide, running a slower chemistry on stored fuel, every membrane still sealed, every gradient still held, waiting for water it could breathe. The brine shrimp cyst, every molecule locked in position inside a glass. Nothing moved, and nothing needed to, because nothing was lost. The rotifer, after 24,000 years, DNA broken in places by 24,000 years of background radiation, and every fragment still present, and the repair enzymes still folded and sitting exactly where they'd be needed. The carp under the ice. Enzymes switched off with phosphate groups, ion gradients holding, because the membranes were sealed against leaks. Self-destruct programs suppressed, so nothing dismantled itself while it waited. Every switch flipped and no wire cut. Now do the opposite. Take the cases where something actually ended and ask what happened physically. At the cellular level, the clearest case is necrosis. The energy supply fails, the pumps stop, water floods in, the membrane stretches past its limit and tears. And in that instant, the compartments merge. Lysosomes, which are essentially chemical demolition units the cell keeps sealed off from everything else, spill their contents. Enzymes that spent the cell's whole life carefully separated from their substrates meet them. Ion gradients that took continuous energy to maintain collapse into a uniform soup. Proteins that were held apart come into contact and aggregate. The word for all of that is mixing. And mixing is the one thing on this entire list that can't be undone by changing the conditions. You can unfreeze something, warm it up. You can undry something, add water. You can unsuffocate something, supply oxygen. Every one of those is a condition you can reverse from the outside, which is precisely why an animal that survives them, survives them. But once the contents of a cell have been stirred together, there is no external condition you can apply that will separate them again. The information wasn't stored in any one molecule. It was stored in which molecules were where. And that's gone the moment the walls come down. So here's what survives contact with all of them. Death is the irreversible loss of the arrangement that lets the process restart. Look at what's inside that sentence and what isn't. Metabolism isn't in it. Oxygen isn't in it. An intact body isn't in it. Time isn't in it. Nor age, nor a lifespan, nor a mortality curve. Cell death isn't in it since the body runs on cell death. All of those turned out to be contingent, local, and mostly descriptions of the way one particular kind of animal is built. What's in it is organization, compartments, gradients, the specific placement of specific molecules with respect to each other. I want to spend a minute on that word arrangement because there's a common assumption that gets in the way, and it's the assumption that all the information in a living thing is in its DNA. It isn't, and the clearest demonstration of that is something you can see in your own cells. Every membrane in your body is physically descended from another membrane. Cells don't manufacture membranes from scratch by reading a gene. They grow them by inserting new lipids and proteins into membranes that already exist. Every membrane in every cell of your body traces back through an unbroken physical chain to the membrane of the egg you came from, and through that egg to your mother's cells and back without a break for as long as there have been cells. The genome specifies what goes into a membrane. It doesn't specify a membrane because a membrane is a structure that only comes from another structure. There's a lovely old experiment that makes the point uncomfortably concrete. In the 1960s, working with Paramecium, a single-celled pond organism covered in rows of cilia, researchers surgically removed a patch of the surface and grafted it back in the reverse orientation. The reversed patch stayed reversed, and when the cell divided, the daughters had it too. Generation after generation, an inherited feature with no change to any gene, carried entirely in the physical geometry of the cell surface, copied because structure was templating structure. That's what I mean by arrangement. It's a real thing. It carries real information, and it's the thing that survives in a brine shrimp cyst and is lost when a cell bursts. Now, for the part I can't tidy up, and I'd rather leave it untidy than pretend. The load-bearing word in that definition is irreversible. And irreversible always has a hidden second half. Irreversible with respect to what? The dried cyst is unrecoverable in a dry room and perfectly recoverable in a puddle. The rotifer in the permafrost is unrecoverable while the ground stays frozen and fine within an hour of a thaw. The carp in the anoxic pond is unrecoverable if it's a trout and unremarkable if it's a carp, in the same water, on the same day. The line between recoverable and not is drawn by the relationship between a state and a set of available conditions. And when the conditions change, the line moves, which means the point of no return isn't a location. It's a boundary that depends on what's available, and what's available includes physics, chemistry, temperature, time, and increasingly, us. Medicine ran into this from the other direction and had to adjust. In severe hypothermia, the chemistry of the body slows so profoundly that the interval before damage becomes unrecoverable stretches out well beyond what it would be at normal body temperature, and people have been brought back from cardiac arrest after periods that would have been hopeless in a warm body. Emergency clinicians have a blunt phrase for the principle involved, about a person not being counted as dead until they've been warmed first. I'm describing a clinical concept here and nothing else. The practice belongs to people trained for it. What matters for our purposes is what the concept reveals. The temperature changed, and the line moved. Nothing about the patient's biology was different from what it had always been. What was different was the range of conditions the process could be restarted from. It shows up in the laboratory too, in a technique lifted more or less wholesale from the brine shrimp. Human embryos are routinely vitrified for storage, which means loaded with cryoprotectants and cooled so fast that the water never gets a chance to crystallize and instead sets into a glass. Molecules immobilized in place, no ice, no mixing, arrangement preserved. Warm it correctly and development resumes from exactly where it stopped. That works because the arrangement was never disturbed. And it's the identical physical principle running in an Artemia cyst on a dry salt pan, arrived at by evolution a very long time before anyone had a freezer. Where it fails is where you'd expect it to fail. Whole organs are enormously harder because a large piece of tissue can't be cooled evenly or fast enough, and the parts that crystallize get their arrangement destroyed. The obstacle there is a phase change and a problem of geometry, and no amount of wanting it badly has moved it much. And there's a final honesty I owe you about this definition, which is that it's harder to apply than it is to state. We have no instrument that measures whether an arrangement is still recoverable. None. We can measure heart rate, brain activity, oxygen, temperature, electrical potentials, metabolic markers, every one of which is a proxy that's already failed us at least once in this script. To find out whether the arrangement was intact, we currently have exactly one method available. Put the thing in the right conditions and see whether it starts, which makes the surviving definition of death retrospective. You can only apply it after the fact. Every case in this script was resolved the same way, by someone adding water or warmth or oxygen and watching. I find that clarifying rather than frustrating because it explains the entire history of this subject. Every one of those animals looked dead to somebody, by every criterion that somebody had, and turned out not to be. And in each case, the thing that settled it was somebody putting it back in the water. So the line is real, and it's sharp, and it's in a very different place from where we've been drawing it. We draw ours where the process stops: heart, breath, brain, the visible signs of activity, the ones our species can detect from across a room and has been detecting for as long as there have been people. That line is enormously useful. It works for us because our arrangement collapses within minutes of the process stopping, so the two events happen close enough together that treating them as one has almost never caused a problem. In the rest of the animal kingdom, those two events come apart and the gap between them is huge. Everything we've looked at lives inside that gap. The medusa reversing its life cycle is inside it. The arm crawling across the reef is inside it. The muscle clamped shut against a dead tide. The cell line that has outlived every animal it ever occupied. The parasite with no mitochondrial genome. The cyst on the salt pan, the rotifer in the permafrost. The clam that had been filtering the same patch of Icelandic mud since before the Reformation. All of them, in the space between stopped and scrambled. None of them cheated anything. Not one. There's no rule being broken anywhere in this script, and no exception being made, and nothing about any of these animals is impossible. What each of them does is stand on a line we drew and demonstrate that we drew it in the wrong place, because we drew it around ourselves and then assumed it went all the way around the world. Before I give you the last of it, I want something from you. And it's a real question with a real disagreement inside it. Somewhere in the last couple of hours, one of these animals crossed a line for you. Maybe it was the brine shrimp cyst with no measurable chemistry, and you thought, that's a seed. That's not an animal in suspension. Maybe it was the rotifer at 24,000 years. Maybe it was the sea star arm and the question of which one of the two animals was the original. Maybe you got all the way to the end and you think the arrangement definition is too generous or not generous enough. Tell me which one, and tell me where you'd put the line. Not what you think the correct scientific answer is, and don't hedge it for me. I want the one where your gut refused to go along with it, because that's the interesting data. I go through the comments on these, and the disagreements down there are usually sharper than anything I could have written up here. If you'd like more of this kind of thing, subscribing is the honest way to say so. And a like takes a second and tells me the long ones are worth the work. Say the word in the comments and I'll go after the other half of this subject, the animals that do the opposite, the ones that die deliberately, on cue, in ways that make no sense at all until you find out what they're paying for. And that's where this lands. Death is the loss of an arrangement, past the point where any available conditions can restore it. That's the whole of it. The stillness, the cold, the silence, the stopped chemistry, the finished years, every signal we have ever used to recognize it turns out to be a symptom that usually travels with the real thing, and in the ocean, routinely, doesn't. The definition that survives contact with all of them is much narrower than the one we use. And it's narrow enough that most of what we call death, out there, is just an animal waiting for better conditions.